Conjugation. 1. Who discovered bacterial conjugation and when? Joshua Lederberg and Edward Tatum discovered bacterial conjugation in 1946. 2. What is bacterial conjugation? It is the process where bacteria transfer DNA through a bridge-like connection. 3. What type of bacteria did Lederberg and Tatum use in their experiment? They used wild-type Escherichia coli. 4. How were mutant strains of E. coli produced in the experiment? They irradiated E. coli with X-rays to induce mutations. 5. What was concluded from Lederberg and Tatum's experiment? Genetic recombination occurred, producing offspring with the wild-type characteristics. 6. What is the role of the donor and recipient bacteria in conjugation? The donor provides DNA, and the recipient receives it. 7. What genetic material is involved in bacterial conjugation? Plasmids and, in some cases, parts of chromosomal DNA. Transduction. 8. What is transduction? It is a genetic transfer process in bacteria facilitated by bacteriophages. 9. What role do bacteriophages play in transduction? They inject their genetic material into bacteria, transferring DNA. Transformation. 10. What is transformation in bacteria? It is the absorption of DNA fragments from the environment by a bacterial cell. 11. What are donor cells in transformation? Bacteria that release DNA fragments into the environment. 12. What happens to a bacterial cell after transformation? It is transformed into a new type of cell. Mutations and genetic recombination in bacteria. 13. What are mutations in bacteria? Changes in bacterial DNA due to replication errors or mutagens. 14. What is stress-directed mutation? A process where genes involved in growth-limiting processes have an increased mutation rate. 15. What are the sources of genetic changes in bacteria? Random mutations and genetic recombination. Phases of bacterial growth. 16. What are the phases of bacterial growth? Lag phase, log phase, stationary phase, and death phase. 17. How long does the lag phase last in E. coli? 10 minutes. 18. What is the bacterial count during the log phase? Approximately 1000 bacteria. 19. What happens in the stationary phase? Bacterial growth stabilizes, reaching 3000 bacteria. 20. How many bacteria remain in the death phase? About 300 bacteria. Nutrient recycling. 21. What role do bacteria play in nutrient recycling? They recycle nutrients like nitrogen, carbon dioxide, and ammonia. 22. Which bacteria fix nitrogen in legumes? Rhizobium bacteria. 23. What is denitrification? The conversion of nitrates into nitrogen gas by denitrifying bacteria. Ecological importance of bacteria. 24. What is the ecological role of bacteria? They decompose organic material, forming humus and enriching soil. 25. What is humus? A nutrient-rich substance formed by bacterial decomposition of dead plants and animals. Economic importance of bacteria. 26. How do bacteria help in digestion? They break down fats, cellulose, and starch. 27. Which vitamin is synthesized by E. coli? Vitamin K and vitamin B12. 28. What are some industrial products made using bacteria? Acetic acid, lactic acid, butanol, and antibiotics like streptomycin. 29. How are bacteria used in food industries? They are used to produce yogurt, cheese, and butter. 30. What is single-cell protein? Protein produced from microorganisms like bacteria, yeast, and fungi. 31. What role do bacteria play in antibiotic production? Bacteria like actinomycetes produce antibiotics such as streptomycin. 32. How do bacteria contribute to biogas production? They decompose organic waste, producing methane gas. 33. What is bioremediation? The use of bacteria to clean up pollutants, like oil spills. 34. How are bacteria used in biotechnology? They produce hormones, vaccines, and genetically engineered products. 35. Which bacteria are used to protect plants from frost? Genetically modified, frost-minus, bacteria. 36. How do bacteria assist in mineral processing? They extract metals like copper, uranium, and gold from low-grade sources. Research applications. 37. What discovery did Hershey and Chase make using bacteria? They proved that DNA is the hereditary material using T2 bacteriophage. 38. How are bacteria used in genetic engineering? To clone genes and produce products like insulin and growth hormones. Thanks for watching. Please like, share, and subscribe.